Hello, everyone. Welcome back to our third and final section of our lymphatics chapter. Today, we're going to be talking about lymph nodes. So lymphatic nodules are collections of lymphatic cells that have a little bit of extracellular matrix, but they do not have a coating of connective tissue surrounding them. If you get collections of lymphatic cells that are surrounded by connective tissue, that's called a lymphatic organ. If you do not have that connective tissue surrounding it, you have a lymphatic nodule. So these collections of lymphatic cells are called germinal centers, and in the middle of the germinal centers, you're going to have B cells and macrophages. So these are the cells that once they become activated, they make antibodies. Macrophages are the big eaters. So those two cell types are at the center of a germinal center. And around that, you're going to have like a candy coating layer of T cells. So these lymphatic nodules, B cells, macrophages surrounded by T cells, their job is to look for antigen and then take action when they find it. Now, there are many types of lymphatic nodules, and one of the most important ones that we're going to talk about are called MALTs, M-A-L-T-S, right? Mucosa-associated lymphatic nodules. So you're going to find these in the various mucosal membranes of the body, including in the GI tract, lining our intestines, the respiratory tract, and the reproductive system, and in the urinary system. All these are areas that are susceptible to microbial attack, so we need special lymphatic tissues to protect us from that kind of invasion. Respiratory, gastrointestinal, genital urinary tracts, these are all major ways that pathogens try to enter our body and make us sick. So these nodules are just going to monitor the area and they will respond to an infection. Now, one particular kind of MALT that we'll be talking about is called a Pyres Patch. And a Pyres Patch is one specific type of MALT which is found in the small intestine. So this word here, ileum, your ileum is a section of your small intestine. So a Pyres Patch is a MALT in the small intestine. MALTs are made of lymphatic nodules, and lymphatic nodules are simply masses of lymphatic cells that do not have a connective tissue capsule. What are they specifically? Macrophages and B cells surrounded by T cells, looking for antigen and defending us. So this is a picture of a Pyres patch. So what we have here is the simple columnar epithelium lining the intestines. So we eat our food, it's chemically broken down in the stomach, and then it passes through the small intestine. We have a simple squamous epithelium because we need to absorb all those nutrients. Well, this is the lumen of the small intestine. Here is the whole rest of the body. Now, what do you have inside intestines, right? We eat food. What comes out the other end is fecal matter, poop. An interesting fact is the majority of fecal matter is not undigested food. In fact, 80% of the dry weight, not including water, 80% of the dry weight of fecal matter is bacteria. So most of what's coming out of your back end is actually bacteria and not undigested food. So that is a lot of bacteria. And it actually has a very important effect on our health, but that's a subject for another class. However, a certain small percentage of the bacteria in that vast population can make you really, really sick. So you have a near endless source of bacteria, some of which may be pathogenic. And the only thing you have separating from the inside where all this bacteria is and the whole rest of your body is an incredibly thin membrane. Remember, that membrane has to be thin because the job of the intestines is to absorb nutrients. If it's really, really thick, it's not going to be able to do its job to absorb nutrients. It must be thin. If it's so thin, that means it's easy for bacteria to penetrate it. Say compared to our skin. Remember our skin with our keratinized, stratified, squamous epithelium, all those layers of dead cells piled on top of each other, a very effective border. But it would also keep out any sort of nutrients. So we need extra protection here in our intestines. And one of the ways that this happens is right here. Here is our Pyres Patch. You can see one germinal center right here in the center, B cells, macrophages, surrounded by T cells. So the job of this structure right here is to keep the bad bacteria in the lumen of your intestine and not penetrating into the rest of your body. Tonsils, right? Those things in the back of your throat are lymphatic nodules. So they are collections of lymphatic tissues, right? You're going to have, they're mostly located in the pharynx. This is the space behind your nasal cavity and your oral cavity. I'll show you a picture in a second. And tonsils, though they look like just smooth spheres, they actually have little holes or pockets, which are called crypts. And what this does, it increases the surface area of the tonsil and allows for antigen to enter and to be detected by many more different types of cells. So the crypts allow for greater exposure of antigen to the various lymphocytes. So here are our three sets of tonsils. We have the pharyngeal tonsils, which are in the nasopharynx. So this is your nose. If you take your finger and put that about 4 or 5 inches back, you will hit the pharyngeal tonsils. Now, when people normally think of tonsils, I think of those two little spheres at the back of their throat. Those are the palatine tonsils because they're next to your palate, right? Your soft palate is the thing that goes up and down the back of your throat. We also have lingual tonsils, which we usually don't see because they're at the root. They're in the very back of our tongue, so you don't see them. So 3 sets of tonsils. What are they? They're just lymphatic nodules. Well, what are lymphatic nodules? They are B cells and T cells, excuse me, B cells and macrophages surrounded by T cells. So that's one lymphatic nodule, here's another lymphatic nodule, a third, a fourth, a fifth, a sixth over here. And these little channels, right, those are the crypts that allow saliva and mucus to kind of percolate through the tonsil and allow these lymphatic cells to detect antigen. So what really the tonsils are, it's kind of a frontline defense at detecting antigen that's going to be coming in by your airway and by coming in through food. So it's just a way to help clean and regulate the material that's entering inside the body. Now, I hesitate to share this, but a disgusting fun fact, these crypts can actually fill up with undigested food and bacteria. And if you want to see what that looks like, just do a YouTube search for Tonsil Stones. I warn you, it's really gross, but there you go. If you want to check it out, you've been warned. Interesting fact though is if somebody has like chronic bad breath, right? If you brush your teeth and then literally 5 minutes later you smell your breath and it stinks, you probably have tonsil stones, which is basically this little space inside the tonsils fills up with undigested food and bacteria, and it doesn't smell very nice. And usually the only way to deal with that is to express them, and you will see that in the tonsil stone videos, should you choose to look it up. Obviously not required. All right, so I should point out that the pharyngeal tonsils are also sometimes called the adenoids. That's sort of the older term for it, right? Palatine tonsils, those little spheres at the back of your throat, and the lingual tonsils at the base of the tongue. Okay, shifting gears a little bit. We're done talking about lymphatic nodules. Now we need to talk about lymphatic organs. When you see that organ, that tells you that you will have a connective tissue capsule surrounding these lymphatic cells. The big lymphatic organs are the thymus, remember that's where T cells go to mature, lymph nodes, and the spleen, which we talked about a little bit earlier, and you remember it had something to do with filtering blood. So the thymus is a bilobed organ. It looks a little bit like a butterfly, right? A thymus, right? Don't confuse thyroid with thymus. Your thyroid is up here in front of your voice box, or above your voice box. Your thymus is anterior to your heart, right? Don't confuse those two organs. If you ever heard of sweetbreads, if you ever go to a fancy restaurant and you see sweetbreads and you're looking for like, you know, a tasty dessert, no. Sweetbreads is thymus of an animal. So just so you ever have that experience, if you order sweetbreads and you get a plate of sort of like raw looking meat, that's what it is. It's thymus. Anyway, back to lecture. So as I mentioned, we have two lobes. Each lobe has an inner and outer layer. The outer layer is called cortex. The inner layer is called medulla. And this organ grows with the body up until puberty. And after puberty, what will happen is all those T cells that are leaving the thymus causes the organ to contract. So I mentioned this in the section on T cells, that after puberty, the thymus actually shrinks in size. So it's one of the few organs that actually gets smaller with age. So here it is. In a child, you see the thymus is huge. It's covering a good deal of the heart. If we look at the histology, now we can see that outer capsule, right? That is connective tissue. Deep to the connective tissue, we'll have lymphatic nodules, right? Just germinal centers, right? They're going to have cortex and medulla, so it's going to be the same situation. Macrophages and B cells surrounded by T cells. So the thymus is the site for immunocompetence of T cells. It's where they become mature. In the cortex is where you will find immature T cells. And as those mature cells, immature cells develop with the help of the thymic nerve cells, they will move towards the medulla. And the medulla of the thymus is where you find mature T cells. And you will find blood vessels associated with the thymic medulla. And when they're ready, they hop from the medulla into the blood vessel and take a trip to some other lymphatic tissue. There they take up residence and search for antigen. Okay. Changing gears, lymph nodes. Lymph nodes, remember a lymph node is a lymphatic organ. It does have a surrounding of, or it does have connective tissue surrounding it. They can vary in size anywhere from 1 to even up to 30 millimeters. So 30 millimeters is a really, really, really big one. If you find one that big, usually the doctor, she's going to want to check it out and make sure there's nothing kind of funny going on. You don't have any lymphoma developing or something like that. But they tend to be relatively small and they're just a little structure encased in connective tissue that holds macrophages, B cells, surrounded by T cells, searching for antigen. And remember that all those lymphatic vessels, which are draining the lymphatic capillaries, those lymphatic vessels drain into lymph nodes. They're bringing the lymph. If you have an infection out in your tissue, right, little components of those pathogens are going to be released into the interstitial fluid. That's swept up into the lymphatic capillaries, and that antigen, right, that evidence of pathogen is then carried in the lymph to the lymph nodes where the various lymphatic cells are waiting. So we have some major groupings of lymph nodes in the body: axillary, inguinal, and cervical. So axillary, armpit; inguinal, groin; cervical is in your neck. So oftentimes when you get sick, that's why you get swelling. That's where you get pain in this area, because you're having activation of all those lymphatic cells in the lymph nodes, and they swell, they get big, right? You can actually feel the cervical lymph nodes get larger in size because of that, what's called clonal expansion. Remember when we went back and looked at what T cells do, right? They undergo mitosis, they increase in numbers. Well, that's why those glands swell. Okay, so lymph nodes have a tough outer connective tissue capsule. The capsule actually dives down and subdivides the lymph node, and that material of the capsule is the same as these subdividing structures, which are called trabecula. In between the trabecula, you will find the lymphatic cells. Now, I'll get to the picture in a second. Lymph nodes are going to have an outer cortex and inner medulla. In the cortex, you're going to have space for lymph to flow. Those are going to be cortical sinuses. Cortical sinuses are just space for the lymph to percolate and flow through. In the medulla, you will have medullary sinuses, just channels or spaces through which lymph can flow through the medulla. Medullary cords refers to just masses of cells in the medulla. So just clumps of cells put together, those are called medullary cords. You will typically have multiple afferent lymphatic vessels. So many vessels, many lymphatic vessels are going to be bringing in lymph to one lymph node. And typically there's only one or two efferent lymphatic vessels that drains fluid out. So what you're getting is like a bunch of in and only one tube out. So you're going to get a buildup of fluid to give those cells enough time to detect any antigen that's coming in from the interstitial fluid. All right, let's cut to the picture. This whole thing is the lymph node. It is covered in a capsule, connective tissue. That capsule, part of it subdivides the lymph node to make the trabecula, shown here. Out here is the cortex. In here is the medulla. Take a look at the cortex. You can see germinal centers. Macrophages, B cells, surrounded by T cells, right? You've heard it over and over again. It's the same basic thing. We're just seeing slight structural differences depending where we are in the body. The spaces around the germinal centers through which lymph flows are the cortical sinuses. When you get deep inside, when you get to the medulla, it has a slightly different structure, slightly different look to it, which is why it has a different name. The spaces of the medulla are called medullary sinuses. The place where you have cells clumped together in the medulla are called medullary cords. Notice that we have multiple afferent lymphatic vessels bringing in lymph from surrounding tissues, but only one efferent lymphatic vessel that's causing lymph to flow away. So we have a pooling, we have kind of like a trap that's collecting all this lymph, which may be carrying antigen if there's some nasty bacteria or pathogen way out in the tissues. What this does is the cells stay put and the antigen comes to them. That's the advantage. As the lymphatic system, as the lymphatic vessels drain lymph from the tissue, that lymph carrying the antigen is brought to the lymphocytes. The lymphocytes don't have to go out wandering if they don't have to. And you might say, okay, why do we really care about that? And part of that is, this is a very efficient system. If we were to have enough lymphatic cells spread evenly throughout our bodies to protect us from infection, we would literally triple in size. It would take that many cells to keep us safe. With this configuration, the cells stay put. When there's a signal of infection, right, the lymph brings it to these cells, they become activated, then they go out and deal with the problem. So it saves a tremendous amount of space and really makes organisms like us possible. Or we're not dominated, we're not 2/3 of our mass isn't lymphatic tissues to protect us. So it may take a little bit of time to coordinate, but it's a huge space saver, so it's a really kind of important concept. Right, here is what a lymph node looks like. Just, you can see multiple afferent vessels. It's kind of hard to see the efferent one from the afferent ones, but sometimes you can tell because you see this concave structure here. This is called a hilum. Lots of organs are going to have a concave structure through which tubes enter or leave. That concave structure through which tubes enter and leave is called a hilum. Here in the lymph node, it's the hilum of the lymph node. We will see other hilums in the spleen and in the liver and in other organs. Okay, so this is just the histology of a lymph node. Out here we have our capsule, we have a bit of a trabecula. Here you can see the germinal centers in the cortex, macrophages and B cells surrounded by T cells. If you have little spaces in the cortex, those are cortical sinuses. The medulla looks substantially different, so masses of cells. The blue stuff, medullary cords. Right, the space in between, medullary sinuses. Okay, let's talk about the spleen. So the spleen is the largest organ associated with the lymphatic system, right? It's responsible for many things, but one of them is for searching for antigens in your blood. If you have bacteria floating around your blood, right, the spleen is looking for antigen. So that means that we need to have these lymphatic cells have access to the blood supply. So that will help you understand some of the structure we're about ready to look at. The spleen also serves as a reservoir for red blood cells and platelets. So if you need extra blood, you're having a sympathetic response, you're bleeding out, you're having some sort of problem, you need extra red blood cells, they're going to be released from the spleen. Now, this is why if you get in a car accident or if you have trauma to your spleen, it can be very, very serious, but because it stores blood, you have the capability of bleeding out very quickly if you have damage to your spleen. Now, also, if you lose part or all of your spleen, it doesn't kill you necessarily, but it makes you much more susceptible to infection and much more affected by having traumatic blood loss. As discussed earlier in the blood section, the spleen is also important for removing old and broken red blood cells, so phagocytizing old dead erythrocytes and platelets, and clearing any bacteria that happen to be in your blood, right? Phagocytizing bacteria and other foreign materials. So look for antigen in the blood, serve as a reservoir for red blood cells, remove and clear out old dead red blood cells, and clear out any pathogen that happens to be inside the blood. So as I mentioned, the spleen is the largest organ of the lymphatic system. On the left side of the body, it is fed by the splenic artery. Remember, that is a branch off the celiac trunk. It is drained by a splenic vein. Both the splenic artery and the splenic vein are going to enter the spleen at its hilum, just a concave structure. It is a lymphatic organ, so it has a capsule. And that same capsular material subdivides the spleen, right? Those subdividing structures are called trabecula. So gross anatomy, this is the diaphragm. This is the left side of the body. Concave structure of the spleen. Notice that it has a connective tissue capsule. This concave structure is the hilum through which splenic artery and splenic vein enter. Remember, the splenic vein is drained by the hepatic portal vein, or drains into the hepatic portal system. Right, this is just, you can see part of the stomach removed, that's the pancreas. This is the diaphragm, right there is the spleen. Right, celiac trunk, splenic artery, splenic vein draining into the liver, which has been partially cut. So trabecular vessels are going to subdivide, trabecular vessels are found inside the spaces subdivided by the trabecula. So you're going to have arteries, trabecular arteries and trabecular veins. And you're going to have two different kinds of pulp. You're going to have white pulp and red pulp. White pulp is associated with trabecular arteries, and red pulp is going to be associated with trabecular veins. What type of cells do you think you're going to find in white pulp? You'll find white cells in white pulp. What kind of cells are you going to find in red pulp? You're going to find red blood cells. So remember, job of the spleen, clean the blood. So incoming blood from arteries is going to get cleaned by white cells in white pulp. The spleen also serves as a blood reservoir. So if you need to add a bunch of red blood cells, you don't want to try to force those through a wall of an arteriole because it's too thick. If blood cells are going to be entering and leaving the circulatory system, they're going to want to do it in veins. Remember the post-capillary venules where you get things like diapedesis. So what am I getting at? Let's cut to the picture. Here we have our trabecular artery feeding one region of white pulp. So this is arterial blood coming into the spleen to be cleaned of antigen and pathogens. So the white pulp is filled with white cells, which are cleaning that blood. On the other side, we have the splenic veins, which are draining these nodules, and they're associated with the red pulp. What do you find in red pulp? In these structures, you're going to find all those stored red blood cells that are waiting to enter the bloodstream when they're called upon. So if you look here, right here at the very center, you're going to have a central artery surrounded by a germinal center, B cells, macrophages surrounded by T cells. Outside of that, you're going to have red pulp where it's associated with splenic veins for the storage of red blood cells that can be added to the circulatory system should the need arise. So this just says in words where I already talked about, white pulp is associated with the T and B lymphocytes and macrophages. you will find in the center of the white pulp, you will find a central artery, just bringing in blood to be cleaned by these lymphocytes. Red pulp is associated with the veins. The red pulp, you will find splenic cords and splenic sinusoids. Splenic cords, this is just structures containing cells. The splenic sinusoids are spaces that are going to contain the stored erythrocytes, stored platelets, and a few other immune cells, but don't worry too much about that. White pulp is for white cells, red pulp is for red cells. Let's just keep it simple like that. Right. Blood can easily enter and leave the bloodstream in the spleen because the capillaries are sinusoidal. So remember that we had continuous, we had fenestrated, and sinusoidal capillaries. So anywhere you would have a large number of cells entering and leaving the spleen, sinusoidal capillaries. So the first place we mentioned was in bone marrow, because that's where a lot of cells enter the circulatory system, and here in the spleen, sinusoidal capillaries. And that brings us, excuse me, to the end of our section on the lymphatic system. So let me know if you guys have any questions, concerns, or suggestions, and I will see you next time. Take care.